Namaste to all students. Kushav Bitiya, myself Jaymin, Dr. Khushbu Verma from Faculty of Agriculture and Veterinary Science. Today we are going to discuss about second type of a cell division, which is meiosis division. This session is related to cell biology course of program BTech FBT and BSc FST second sem. So meiosis process is generally present only in gamete cell formation. It not happen during the somatic cell division. Meiosis division was discovered and described for the first time in sea urchin eggs in 1876 by German biologist Oscar Hardwick. It was described again in 1883 at the level of chromosomes by the Belgian zoologist Eduard van Beneden in Ascaris worm's egg. The term meiosis was coined by the J.B. Farmer and J.B. Murray in 1905. This meiosis process is basically where it occurs. So meiosis process occurs only in eukaryotic cell cycle, life cycles involving sexual reproduction. It always occurs in reproductive cells, means meiocytes, and in lower plants, meiosis occurs after fertilization in the zygote, and in higher plants, it occurs before fertilization in the time of gamete creation. So this is the basic difference in between high plants and lower plants, and the timing of the meiosis, because in lower plants meiosis happens after fertilization, and in higher plants it occurs before fertilization during the gamete formation. Now we can discuss about some facts which are very much important about meiosis process. So meiosis is the type of cell division by which germ cells are produced. Two meiotic divisions are basically present, meiosis 1 and meiosis 2. Meiosis 1 is original meiosis division because in this stage actually cell gives or derive their half number of chromosomes, because in meiosis process after the completion process you will get 4 daughter cells, and but number of chromosomes will become half in comparison to parental cell. If you are talking about that the parental cell is 2n in nature, means diploid, so 4 daughter cells produce haploids 1n chromosome 1n cell. Now comes the daughter cells which contain half the number of chromosomes as the original cell. During this process, meiosis process, gamete formation happens, means egg and sperm produces. It occurs in the testis in males in the form of spermatogenesis, and it also occurs in the ovaries in female, known as oogenesis. During meiosis, DNA replicates once, but the nucleus divides twice. Now come to the detailed discussion about the meiosis 1 and meiosis 2. Meiosis 1 is the reduction division where diploid mother cell produces 2 haploid cells. In meiosis 1, there are 4 categories: prophase 1, metaphase 1, anaphase 1, and telophase 1. Prophase 1 again subdivided into five members like leptotene, zygotine, pachytene, diplotin, and dikinesis. Now come to the prophase 1 stage. During prophase 1, DNA is exchanged between homologous chromosomes in a process called homologous recombination. This often results in chromosomal crossover. The paired and replicated chromosomes are called bivalent or tetrads. The process of pairing the homologous chromosomes is called synapsis, and at this stage, non-sister chromatids may cross over at points called chiasmata, and the singular name is chiasma. In prophase 1, first stage is leptotene, and in the leptotene, This is also known as leptonema from Greek word meaning thin threads. During this stage, individual chromosomes begin to condense into long strands within the nucleus. However, the two sister chromatids are still so tightly bound that they are indistinguishable from one another. Next substage of prophase 1 is zygote. The zygote stage, also known as zygonema, from Greek word meaning paired threads. Zygote occurs as the chromosome approximately line up with each other into homologous chromosomes. The combined homologous chromosomes are said to be bivalent. Third stage is pachytene of meiosis 1. So in pachynema, which is synonym of this name, so in pachynema, the homologous chromosomes become much more closely associated. This process is also known as synapsis. The synapsed homologous pairs of chromosomes is called a tetrad because it consists of four chromatids. It can't be observed until the next stage, but the synapsed chromosomes may undergo the crossing over in pachytene stage. The chromosome continues to condense. Here you can understand this all key events which are happening during this stage more easily. This is pachytene stage of cell where you can simply see two homologous chromosomes are paired along with each other and their sister chromatids are arranged very closely to each other so that they can cross over, they can exchange their genetic material, and this happens by the help of synapses in which two homologous chromosomes paired with each other and they exchange their genetic material information. And when after the completion of genetic material exchange, these two chromosomes which are right now having pair, so they start to separate from each other, and by the extension of this tension on this synapses, a structure becomes visualized is known as chiasma. And during the pachytene stage, chromosome continue to condense. Next one is the diplotin stage of prophase 1. So during the diplotin stage, also known as diplonema, from Greek word meaning two threads, the homologous chromosomes in diplotin stage during the homologous chromosomes separate from each other a little and the chromosome themselves uncoil a bit, allowing some transcription of the DNA molecule. In last stage of prophase 1 is dikinesis. So chromosomes condense further during the dikinesis stage from Greek word meaning moving through. This is the first point in meiosis where four parts of the tetrads are actually visible, and in this stage the homologous chromosomes separate further and the chiasmata terminalize, means chiasmata formation disappears, making chiasmata clearly visible. Here you can understand this different key events of prophase 1. You can simply see in this diagram. This is leptotin, in which chromosomes are arranged in a bouquet form, and in the zygote pairing happens of homologous chromosomes, and they start to do synapses. And in pectine, synapses completed and crossing over happens. And after the completion of crossing over in the diplotin stage, this chiasmata start to appear, because after the completion of synapses and crossing over or exchange material, these homologous chromosomes start to separate from each other, and by the process of separation, this chiasmata structure appear. And in the last stage is which is dikinesis, two homologous chromosomes completely separate from each other. There is a terminalization of synapses happens, because crossing over process has been completed during the pectine stage. So these all are stages which are related for with the prophase 1 stage. Now come to the discussion about crossing over. So during prophase 1 of meiosis, a part of chromatid, sister chromatid of one chromosome goes over to a sister chromatid of other chromosome and vice versa. The interchange of the parts of chromatids of a pair of homologous chromosome is known as crossing over. Crossing over is a process by which two chromosomes of a homologous pair exchange equal segment with each other, and crossing over occurs in the first division of the meiosis. Means crossing over only present in meiosis 1, and especially in meiosis 1 it is present in prophase 1 stage, sub-stage, which is pectine. Here you can understand about the crossing over with the more understanding points with this diagram, where you can simply see these two are, this one is sister chromatid from one chromosome, and this red highlighted is another sister chromatid from another chromosome. They come close together and they create the synapses. They exchange their genetic material information. Now this chromatid is having this region, and this chromatid is having this region. During the synapses, two homologous chromosomes start to pair, and after the pairing, means synapses, they do crossing over, they exchange their genetic material information. What is the importance of crossing over? Why we need crossing over during the meiosis process? So crossing over takes place between the non-sister chromatids, means one sister chromatid from another chromosome, and second sister chromatid from next second one chromosome. So crossing over is one of the most important events in meiosis, because it allows variation in the produced offspring. Basically, it produces new individuals having new combinations of the trait, helped in establishing the concept of linear arrangement of the genes. It helps in the mapping of chromosomes, and it helps in the selection of useful combinations. After the successful completion of prophase 1, cell undergoes to the metaphase 1 stage. So metaphase 1 is the second phase of meiosis, and the tetrads from prophase 1 lined up in the middle of dividing cell randomly. Spindle fibers attached to the tetrad from both end of the cell. Third step of meiosis 1 is anaphase 1. So anaphase 1 begins when two chromosomes of each bivalent separate and start moving towards opposite poles. In anaphase 1, the sister chromatids remain attached at their centromere and move together towards the poles. Means in this stage or in this process, sister chromatids will not be separated from each other, means they will not be separated by the separase enzyme. There is no any use of anaphase promoting complex. So simply complete chromosome, one chromosome will move from towards their pole and second one will move in the opposite direction towards their respective pole. Here you can understand this more by this diagram of the cell, where you can simply see this is the complete chromosome with its complete two sister chromatids. They will move towards their respective poles. These two chromosomes are going to move at this pole and these two are going into the opposite direction. Last stage is telophase 1 of meiosis 1. So the homologous chromosomes pair reach to the poles of the cell. The homologous chromosome pairs complete their migration to the two poles. A nuclear envelope reforms around each chromosome set and the spindle fiber finally disappears and cytokinesis follows. After the completion of cytokinesis, two cells will start to form. Here you can simply see by this diagram, after the completion of telophase 1, two nuclei will be formed, daughter nuclei will be formed, which is having two chromosomes. In parental cell, it was having 4 chromosomes, but now daughter cells are having only 2 chromosomes, mean number of chromosomes have become half after the completion of telophase 1. Next, after the completion of meiosis 1, cell undergoes to the meiosis second. So meiosis second is not a true meiosis division. It behaves like a mitotic division. So meiosis second is basically mitotic division of two haploid cells to produce finally 4 haploid daughter cells. It includes prophase second, metaphase second, anaphase second, and telophase second. In prophase second, meiosis second basically begins without any further replication of the chromosome. In prophase second, the nuclear envelope breaks down and the spindle apparatus forms. While chromosome duplication took place prior to the meiosis 1, no new chromosome replication occurs before meiosis second. The centrioles duplicate. This occurs by the separation of two members of the pair and then the formation of two daughter centriole perpendicular to each original centriole. The two pair of centrioles separate from two centrosomes. The nuclear envelope breaks down and the spindle fiber or spindle apparatus forms in prophase second. In metaphase second, which is the next stage of meiosis second, so the chromosome becomes arranged on metaphase plate. Centromeres are arranged in a line called equatorial plate of invisible spindle apparatus, and previously occurring nuclear membrane is not present in this metaphase stage. In next stage, which is anaphase second of meiosis second, so the centromeres separate and the sister chromatids, now individual chromosomes, move towards the opposite poles of the cell. Means in this stage, means in the anaphase second, sister chromatids will be separated from each other, but in case of anaphase 1, there was no any kind of a separation of sister chromatids. Complete chromosomes were going towards their poles. But in case of anaphase second, sister chromatids will be separated from each other and they will move towards their respective poles. So the separated chromatids are now called chromosomes in their own right. After the successful completion of anaphase second, cell undergoes to the telophase second of meiosis second division. So nuclear envelope again start to form, nucleus reappear and chromosome lengthen and become indistinct. So after the completion of meiosis second, you get four nuclei and cytokinesis happens by which four daughter cells prepared or formed by and along with the four daughter cells they are having half number of the chromosomes in comparison to the parental cell. Here you can understand complete meiosis division by this diagram. So here I'm sharing the slide. Here you can simply see this is the early prophase 1 and next one is the middle prophase second one and this one is the middle prophase 1. After the completion of prophase 1 stage, crossing over happens and genetic material exchange from each other. And this prophase 1 is having 5 sub-stages: leptotene, zygotene, pachytene, diplotene, and dikinesis. In pachytene, crossing over happens, synapsis completion happens during the dikinesis, diplotene and dikinesis process. These chromosomes separate from each other, and in metaphase stage, all chromosomes arrange at a equatorial plate or metaphase plate. In anaphase 1, complete chromosomes will move towards their respective poles. There is no separation of sister chromatids during this replication, this cell division process. And after completion of anaphase 1, cell undergoes to the telophase 1 stage where two nuclei with half number of chromosomes appears. Nuclear membrane reappear and nucleolus reappear during the telophase 1 stage. After completion of telophase 1 stage, cells undergo to the prophase 2nd stage, where again chromosome start to condense and nuclear membrane again disappear, spindle fiber again reform. Now, after the completion of prophase 2nd, cell undergoes to the metaphase 2nd stage, where these chromosomes are arranged at metaphase plate, spindle fibers completely grown up, and after completion of metaphase 2nd, cell undergoes to the anaphase 2nd, where these two sister chromatids means in one chromosome which is having two sister chromatids, so two sister chromatids of one same chromosome, they are separated from each other by the separase enzyme, and now these sister chromatids are moving towards their respective poles. After successful completion of anaphase 2nd, cell undergoes to the telophase 2nd stage, where now four nuclei has been prepared, and after successful completion of telophase 2nd, cell goes into cytokinesis process by which you are getting four daughter cells which are having half number of chromosomes. And this kind of a cell division happens only in gamete cell formation process. And now in the last important points, you have to know about the meiosis process importance. So basically it produces haploid gametes so that the diploid number of the species remain constant generation after generation. Means when gametes form, so they are haploid in nature due to meiosis process. These gametes means male gamete and female gamete further fuse and fertilize. So after the fertilization process, here is one end from male and second end from mother cell, so they become fertilized. So after the fertilization of two gametes, it becomes again diploid zygote and diploid embryo. So basically meiosis helps the species to remain constant generation after generation, means diploid number should be remain, and a source of genetic variation happens only during the meiosis process, and especially in the meiosis 1, which is true meiosis process. And in meiosis 1 it happens only in prophase 1 pachytene stage, where crossing over happens and it brings together new gene combination on chromosomes. So now this is the sum up of this session, and this session is powered by digital version 2.0 Jyoti Vidyapeeth Women's University Jaipur. And if you have any query, then mention in the comment box. I will resolve it. Thank you.